Before we begin, if you're finding peace through these sessions, we'd love for you to take a moment, when you're awake, of course, to leave a review and subscribe on whatever channel you're listening on. Your support helps others discover this space and join our journey to restful sleep. Welcome to Slothful Slumber, the podcast where we transform the vast knowledge of the internet into a gentle lullaby for your mind. I'm your host, Luna Dreamwell, and I'm here to guide you on a journey to tranquility through the soothing cadence of Wikipedia articles. Each episode will delve into the most wonderfully mundane and sleep-inducing topics, helping you drift off into a realm of serene dreams. So find your comfortable space, close your eyes, and let the dull hum of information carry you into a restful slumber. This Wikipedia article is titled Electric Eel. The electric eels are a genus, Electrophorus, of neotropical freshwater fish from South America in the family Gymnotidae. They are known for their ability to stun their prey by generating electricity, delivering shocks at up to 860 volts. Their electrical capabilities were first studied in 1775, contributing to the invention of the electric battery in 1800. Despite their name, electric eels are not closely related to the true eels, anguilliforms, but are members of the electroreceptive knifefish order Gymnotiformes. This order is more closely related to catfish. In 2019, electric eels were split into 3 species. For more than 2 centuries before that, the genus was believed to be monotypic, containing only Electrophorus electricus. They are nocturnal, obligate, air-breathing animals with poor vision complemented by echolocation. They mainly eat fish. Electric eels grow for as long as they live, adding more vertebrae to their spinal column. Males are larger than females. Some captive specimens have lived for over 20 years. Evolution. Taxonomy. When the species now defined as Electrophorus electricus was described by Carl Linnaeus in 1766, based on early field research by Europeans in South America and specimens sent back to Europe for study. He used the name Gymnotus electricus, placing it in the same genus as Gymnotus carapo, the banded knife fish. He noted that the fish is from the rivers of Suriname, that it causes painful shocks, and that it had small pits around the head. In 1864, Theodore Gill moved the electric eel to its own genus, Electrophorus. The name is from the Greek electron, amber, a substance able to hold static electricity, and phero, I carry, giving the meaning electricity bearer. In 1872, Gill decided that the electric eel was sufficiently distinct to have its own family, Electrophoridae. In 1998, Albert and Campos de Paz lumped the Electrophorus genus with the family Gymnotidae, alongside Gymnotus, as did Ferraris and colleagues in 2017. In 2019, C. David de Santana and colleagues divided E. electricus into 3 species based on DNA divergence, ecology and habitat, anatomy and physiology, and electrical ability. The 3 species are E. electricus, now in a narrower sense than before, and the 2 new species, E. voltae and E. varii. However, this revision did not address Electrophorus multivalvulus, which was described from the Peruvian Amazon by Nakashima in 1941. Therefore, E. varii, described from the same region, may be a junior synonym of E. multivalvulus and has been regarded as such by some biologists. Phylogeny. Electric eels form a clade of strongly electric fishes within the order Gymnotiformes, the South American knife fishes. Electric eels are thus not closely related to the true eels, Anguilliformes. The lineage of the Electrophorus genus is estimated to have split from its sister taxon Gymnotus sometime in the Cretaceous. Most knife fishes are weakly electric, capable of active electrolocation, but not of delivering shocks. Their relationships, as shown in the cladogram, were analyzed by sequencing their mitochondrial DNA in 2019. Actively electrolocating fish are marked with a small yellow lightning flash. Fish able to deliver electric shocks are marked with a red lightning flash. Species. There are 3 described species in the genus, not differing significantly in body shape or coloration. Electrophorus electricus, Linnaeus, 1766. This, the type species, has a U-shaped head with a flattened skull and cleithrum. Electrophorus voltae. Specified in 2019. This species is the strongest bioelectricity generator in nature, capable of generating 860 volts. Like E. electricus, this species has a flattened skull and cleithrum, but the head is more egg-shaped. Electrophorus vari, also specified in 2019. Compared to the other 2 species, this one has a thicker skull and cleithrum, but the head shape is more variable. E. varii appears to have diverged from the other species around 7.1 million years ago during the late Miocene, while E. electricus and E. voltae may have split around 3.6 million years ago during the Pliocene. Ecology. The 3 species have largely non-overlapping distributions in the northern part of South America. E. electricus is northern, confined to the Guiana Shield, while E. voltae is southern, ranging from the Brazilian Shield northwards. Both species live in upland waters. E. varii is central, largely in the lowlands. The lowland region of E. varii is a variable environment, with habitats ranging from streams through grassland and ravines to ponds, and large changes in water level between the wet and dry seasons. All live on muddy river bottoms and sometimes swamps, favoring areas in deep shade. They can tolerate water low in oxygen as they swim to the surface to breathe air. Electric eels are mostly nocturnal. E. voltae mainly eats fish, in particular the armored catfish Megalechus thoracata. A specimen of E. voltae had a caecilian, a legless amphibian, Typhlonectes compressicauda in its stomach. It is possible that this means that the species is resistant to the caecilian's toxic skin secretions. E. voltae sometimes hunts in packs and have been observed targeting a shoal of tetras, then herding them and launching joint strikes on the closely packed fish. The other species, A. vari, is also a fish predator. It preys especially on Calycthiidae, armored catfishes, and Cycladae, cichlids. Biology. General biology. Electric eels have long, stout bodies, being somewhat cylindrical at the front, but more flattened towards the tail end. E. electricus can reach 2 meters, 6 feet 7 inches in length, and 20 kilograms, 44 pounds in weight. The mouth is at the front of the snout and opens upwards. They have smooth, thick, brown to black skin with a yellow or red underbelly and no scales. The pectoral fins each possess 8 tiny radial bones at the tip. They have over 100 precaudal vertebrae, excluding the tail, whereas other gymnotids have up to 51 of these. There can be as many as 300 vertebrae in total. There is no clear boundary between the tail fin and the anal fin, which extends much of the length of the body on the underside and has over 400 bony rays. Electric eels rely on the wave-like movements of their elongated anal fin to propel themselves through the water. Electric eels get most of their oxygen by breathing air using buccal pumping. This enables them to live in habitats with widely varying oxygen levels, including streams, swamps, and pools. Uniquely among the gymnotids, the buccal cavity is lined with a frilled mucosa, which has a rich blood supply, enabling gas exchange between the air and the blood. About every 2 minutes, the fish takes in air through the mouth, holds it in the buccal cavity, and expels it through the opercular openings at the sides of the head. Unlike in other air-breathing fish, the tiny gills of electric eels do not ventilate when taking in air. The majority of carbon dioxide produced is expelled through the skin. These fish can survive on land for some hours if their skin is wet enough. Electric eels have small eyes and poor vision. They are capable of hearing via a Weberian apparatus, which consists of tiny bones connecting the inner ear to the swim bladder. All of the vital organs are packed in near the front of the animal, taking up only 20% of space and sequestered from the electric organs. Electrophysiology. Electric eels can locate their prey using electroreceptors derived from the lateral line organ in the head. The lateral line itself is mechanosensory, enabling them to sense water movements created by animals nearby. The lateral line canals are beneath the skin, but their position is visible as lines of pits on the head. Electric eels use their high-frequency sensitive tuberous receptors distributed in patches over the body for hunting other knife fish. Electric eels have 3 pairs of electric organs, arranged longitudinally: the main organ, hunter's organ, and sax organ. These organs give electric eels the ability to generate 2 types of electric organ discharges: low voltage and high voltage. The organs are made of electrocytes, modified from muscle cells. Like muscle cells, the electric eel's electrocytes contain the proteins actin and desmin, but where muscle cell proteins form a dense structure of parallel fibrils, in electrocytes they form a loose network. 5 different forms of desmin occur in electrocytes, compared to 2 or 3 in muscle. But its function in electrocytes remained unknown as of 2017. Potassium channel proteins involved in electric organ discharge, including KCNA1, KCNH6, and KCNJ12, are distributed differently among the 3 electric organs. Most such proteins are most abundant in the main organ and least abundant in Sachs's organ, but KCNH6 is most abundant in Sachs's organ. The main organ and hunter's organ are rich in the protein calmodulin, involved in controlling calcium ion levels. Calmodulin and calcium help to regulate the voltage-gated sodium channels that create the electrical discharge. These organs are also rich in sodium-potassium, ATPase, an ion pump used to create a potential difference across cell membranes. The maximum discharge from the main organ is at least 600 volts, making electric eels the most powerful of all electric fishes. Freshwater fishes like the electric eel require a high voltage to give a strong shock because freshwater has high resistance. Powerful marine electric fishes like the torpedo ray give a shock at much lower voltage, but a far higher current. The electric eel produces its strong discharge extremely rapidly at a rate of as much as 500 hertz, meaning that each shock lasts only about 2 milliseconds. To generate a high voltage, an electric eel stacks some 6,000 electrocytes in series longitudinally in its main organ. The organ contains some 35 such stacks in parallel on each side of the body. The ability to produce high voltage, high frequency pulses, in addition, enables the electric eel to electrolocate rapidly moving prey. The total electric current delivered during each pulse can reach about 1 ampere. It remains unclear why electric eels have 3 electric organs, but basically produce 2 types of discharge, to electrolocate or to stun. In 2021, Junshu and colleagues stated that Hunter's organ produces a 3rd type of discharge at a middle voltage of 38.5 to 56.5 volts. Their measurements indicate that this is produced just once, for less than 2 milliseconds, after the low voltage discharge of Sachs's organ and before the high voltage discharge of the main organ. They believed that this is insufficient to stimulate a response from the prey, so they suggested it may have the function of coordination within the electric eel's body, perhaps by balancing the electrical charge, but state that more research is needed. When an electric eel identifies prey, its brain sends a nerve signal to the electric organ. The nerve cells involved release the neurotransmitter chemical acetylcholine to trigger an electric organ discharge. This opens ion channels, allowing sodium to flow into the electrocytes, reversing the polarity momentarily. The discharge is terminated by an outflow of potassium ions through a separate set of ion channels. By causing a sudden difference in electric potential, it generates an electric current in a manner similar to a battery, in which cells are stacked to produce a desired total voltage output. It has been suggested that Sachs's organ is used for electrolocation. Its discharge is of nearly 10 volts at a frequency of around 25 hertz. The main organ, supported by Hunter's organ in some way, is used to stun prey or to deter predators. It can emit signals at rates of several hundred hertz. Electric eels can concentrate the discharge to stun prey more effectively by curling up and making contact with the prey at 2 points along the body. It has also been suggested that electric eels can control their prey's nervous systems and muscles via electrical pulses, keeping prey from escaping or forcing it to move so they can locate it. But this has been disputed. In self-defense, electric eels have been observed to leap from the water to deliver electric shocks to animals that might pose a threat. The shocks from leaping electric eels are powerful enough to drive away animals as large as horses. Life cycle. Electric eels reproduce during the dry season, from September to December. During this time, male-female pairs are seen in small pools, left behind after water levels drop. The male makes a nest using his saliva, and the female deposits around 1,200 eggs for fertilization. Spawn hatch 7 days later, and mothers keep depositing eggs periodically throughout the breeding season, making them fractional spawners. When they reach 15 millimeters, 0.59 inches, the hatched larvae consume any leftover eggs, and after they reach 9 centimeters, 3.5 inches, they begin to eat other foods. Electric eels are sexually dimorphic, males becoming reproductively active at 1.2 meters in length and growing larger than females. females start to reproduce at a body length of around 70 centimeters, 4 inches. The adults provide prolonged parental care lasting 4 months. E. electricus and E. voltae, the two upland species which live in fast-flowing rivers, appear to make less use of parental care. The male provides protection for both the young and the nest. Captive specimens have sometimes lived for over 20 years. As the fish grow, they continually add more vertebrae to their spinal column. The main organ is the first electric organ to develop, followed by Sachs's organ and then Hunter's organ. All the electric organs are differentiated by the time the body reaches a length of 23 centimeters, 9.1 inches. Electric eels are able to produce electrical discharges when they are as small as 7 centimeters, 2.8 inches. Interactions with humans. Early research. The first written mention of the electric eel or purake, the one that numbs in Tupi, is in records by the Jesuit priest Fernand Cardim in 1583. The naturalists Bertrand Bajon, a French military surgeon in French Guiana, and the Jesuit Ramon M. Termeyer in the River Plate Basin conducted early experiments on the numbing discharges of electric eels in the 1760s. In 1775, the torpedo, the electric ray, was studied by John Walsh. Both fish were dissected by the surgeon and anatomist John Hunter. Hunter informed the Royal Society that Gymnotus electricus appears very much like an eel, but it has none of the specific properties of that fish. He observed that there were 2 pair of these electric organs, a larger, the main organ, and a smaller, Hunter's organ, one being placed on each side, and that they occupied perhaps more than 1/3 of the whole animal by volume. He described the structure of the organs, stacks of electrocytes, as extremely simple and regular, consisting of 2 parts, viz., flat partitions or septa, and cross-divisions between them. He measured the electrocytes as 1/17 inch, 1.5 millimeters, thick in the main organ, and 1/56 inch, 0.45 millimeters, in Hunter's organ. Also, in 1775, the American physician and politician Hugh Williamson, who had studied with Hunter, presented a paper, Experiments and Observations on the Gymnotus electricus, or electric eel, at the Royal Society. He reported a series of experiments, such as, in order to discover whether the eel killed those fish by an emission of the same electrical fluid with which he affected my hand when I had touched him, I put my hand into the water at some distance from the eel. Another catfish was thrown into the water. The eel swam up to it and gave it a shock, by which it instantly turned up its belly and continued motionless. At that very instant, I felt such a sensation in the joints of my fingers as in experiment 4, and instead of putting my hand into the water, at a distance from the eel, as in the last experiment, I touched its tail, so as not to offend it, while my assistant touched its head more roughly. We both received a severe shock. The studies by Williamson, Walsh, and Hunter appear to have influenced the thinking of Luigi Galvani and Alessandro Volta. Galvani founded electrophysiology with research into how electricity makes a frog's leg twitch. Volta began electrochemistry with his invention of the electric battery. In 1800, the explorer Alexander von Humboldt joined a group of indigenous people who went fishing with horses, some 30 of which they chased into the water. The pounding of the horses' hooves, he noted, drove the fish up to 5 feet, 1.5 meters, long out of the mud, and prompted them to attack, rising out of the water and using their electricity to shock the horses. He saw 2 horses stunned by the shocks and then drowned. The electric eels, having given many shocks, now require long rest and plenty of nourishment to replace the loss of galvanic power they have suffered, swam timidly to the bank of the pond, and were easily caught using small harpoons on ropes. Humboldt recorded that the people did not eat the electric organs, and that they feared the fish so much that they would not fish for them in the usual way. In 1839, the chemist Michael Faraday extensively tested the electrical properties of an electric eel imported from Suriname. For a span of 4 months, he measured the electrical impulses produced by the animal by pressing shaped copper paddles and saddles against the specimen. Through this method, he determined and quantified the direction and magnitude of electric current and proved that the animal's impulses were electrical by observing sparks and deflections on a galvanometer. He observed the electric eel increasing the shock by coiling about its prey, the prey fish representing a diameter across the coil. He likened the quantity of electric charge released by the fish to the electricity of a Leyden battery of 15 jars, containing 23,000 square centimeters of glass coated on both sides, charged to its highest degree. The German zoologist Carl Sachs was sent to Latin America by the physiologist Émile Dubois-Reymond to study the electric eel. He took with him a galvanometer and electrodes to measure the fish's electric organ discharge, and used rubber gloves to enable him to catch the fish without being shocked, to the surprise of the local people. He published his research on the fish, including his discovery of what is now called Sachs's organ, in 1877. Artificial electrocytes. The large quantity of electrocytes available in the electric eel enabled biologists to study the voltage-gated sodium channel in molecular detail. The channel is an important mechanism as it serves to trigger muscle contraction in many species, but it is hard to study in muscle as it is found in extremely small amounts. In 2008, Jianshu and David Lavin designed artificial cells that would be able to replicate the electrical behavior of electric eel electrocytes. The artificial electrocytes would use a calculated selection of conductors at nanoscopic scale. Such cells would use ion transport as electrocytes do, with a greater output power density and converting energy more efficiently. They suggest that such artificial electrocytes could be developed as a power source for medical implants, such as retinal prostheses and other microscopic devices. They comment that the work has mapped out changes in the system-level design of the electrocyte that could increase both energy density and energy conversion efficiency. In 2009, they made synthetic protocells, which can provide about a 20th of the energy density of a lead-acid battery and an energy conversion efficiency of 10%. In 2016, Hao Sun and colleagues described a family of electric eel mimicking devices that serve as high-output voltage electrochemical capacitors. These are fabricated as flexible fibers that can be woven into textiles. Sun and colleagues suggest that the storage devices could serve as power sources for products such as electric watches or light-emitting diodes.